Hey everyone, we are starting a new chapter. This is the chapter 2 notes. We are starting our discussion on the cell. If you recall, in chapter 1, we started talking about levels of organization, how we can study life, including humans, at the molecular level, cellular, tissues, organs, organ systems, organisms. In order for us to get to organ systems, to study things like cardiovascular system, respiratory system, reproductive system, we first need to understand the individual components. So that's where we're starting here. We're starting with the cell, the smallest unit of life for all living things, including humans. So in this chapter, we're going to become familiar with different types of microscopy, look at the different organelles of the cell, and of course, since this is anatomy, look at how the structure of parts of a cell relate to their functions, as well as the whole cell. We'll eventually get to cell cycles and differentiation. That'll actually be the next chapter. And in lab, we'll look at how to use a microscope and identify all these things under a microscope. To me, cells are a lot like a factory. This factory here, this assembly line, is showing the making of a car. I'm not really sure what kind of car, but when you manufacture a car, what happens is you have different rooms that do different things in the construction of a car. So first you'll build the framework, the chassis, and then you'll put on the paneling, and then you'll put on the engine block. I don't really know the order, but something like that. And then you put on the paint, and you put in the seats inside or something like that. And then you do the final checks at the end. You go through a whole process, step by step by step by step. This is a lot like what happens inside of a cell. Inside of a cell, you've got tons of processes and different compartments, different places that are specialized to do certain things. We'll talk about, so to emphasize those two words, where can I write it? I guess I'll write it on the next slide because it's too busy here. We'll be talking about compartmentalization. How different compartments, different rooms, so to speak, within a cell help make things more efficient and can do things very specialized and specialized functions as well. Each room, each organelle has a specialized function, specialization. By having compartmentalization and specialization, that makes cells very efficient. To see very small things, again, we're going to focus on this in lab, but to see very small things, we need microscopes. With our own eyes, we can only see, like the largest human cell would be a human egg. And those are just look like tiny dots. To actually see the detail of cells, you need microscopes. And there's different types of microscopes. So let's briefly overview the types of microscopes that we'll be, if not looking at in lab, we'll be looking at pictures at in class. And to point out the unit that we'll be using, so that's not actually the one that we'll be using, to point out the unit that we'll be using mostly in this class, we'll of course use the metric system. That's what is universally accepted in science. Cells are on the micrometer level. This is pronounced micrometer, or I guess in some cases you can say micrometer, but the American way is micrometer. The abbreviation for micrometer is this. Let me rewrite it. This letter here is called mu. It's a Greek letter. And then the lowercase m is for meter, micrometer. A micrometer is a millionth of a meter, really, really, really small. And if you have time, I encourage you to check out this Scale of the Universe site. It gives you an idea of how small, how big, or how small that really is. What we'll be using in lab are compound light microscopes. The word compound means multiple. So it's a compound microscope because it's got multiple lenses. And we'll see how that comes into play. It helps you change magnification. Light, because you're shining light from the bottom of the microscope. As you shine light through the thing you're looking at, through your specimen, it the parts that are thinner will look lighter, and the parts where there's stuff in the way will appear darker. The resolution that we can see with, like, sorry, let me back up. The term resolution is a term to describe how well you can differentiate really small things. The smallest things that we can differentiate are cells and large organelles. I know I haven't really defined that term yet, organelle. An organelle is a structure within cells, and there are different types of organelles. That's what we're going to go over here today. But those are the largest things. So you can see in this picture here, these are not human cells, but you can see 4 cells, 1, 2, 3, 4. And you can see some larger organelles, which I'll color in black. Like these things are called, they're called the nucleus. There are some smaller organelles here, but they're still pretty large that you can see them. Those are the types of things you can see with light microscopes. So there's other stuff in here. There's other stuff in this cell, but you can't see the detail of it. Just like if you were really, really high up in an airplane, you might be able to make out like large landforms and buildings, but if 2 people were right next to each other, you wouldn't be able to tell them apart because you're too high up. You'd have to get closer down to the ground to see that they're in fact 2 people. This is the same idea. We don't have the resolution to make out those really small things with a compound light microscope. So this is what we'll be using most in class because we can see the basic details of cells, but we can't see the really small cellular things, small organelles. In order to do that, we need a specialized microscope. It's called an electron microscope. There are 2 different types of electron microscopes that I want to go over. This first one is called a transmission electron microscope. To transmit means to go through. So I think of it that way because we're looking into the cell. These are these same 4 cells that we saw before, but you can clearly see that while we can see those same large organelles, we also see tons and tons of fine detail. We have much higher resolution. Transmission electron microscopes lets us see inside of the thing we're looking at. So if we're looking at cells, it lets us see inside of cells. If you're curious what it looks like, it's this large device here compared to, I have no idea what size screen this is, but compared to a standard, I don't know what standard is anymore, but compared to a computer screen here, it's a really, really large device. I'm not going to test you on the physics of how all this works, the engineering of it. I don't really fully understand it, but it uses a lot of important technology and it's very expensive. We don't have one of these in our lab, but we'll be looking at pictures, in some pictures here and there where we see transmission electron micrographs to see the high detail of organelles inside of cells. But sometimes you might want to see what's going on the surface. Transmission lets you see inside, but what does the surface of a cell look like? To do that, you need a scanning electron microscope. With a scanning electron microscope, again, I'm not going to test you on how this whole device works. It's also pretty big, but you do coat it. You do coat biological specimens, cells, with a special material, and that allows you to see what's going on the surface of these cells. It's not just a plain covering. There's important structures on the surface, which is what we'll discuss here in a minute, what's found on these plasma membranes. So both types of electron microscopes, both transmission and scanning electron microscopes, they both let us see with really high resolution. The difference is transmission lets us see inside of cells, scanning lets us see on the surface. So here's an example of a cell structure called cilia. We'll talk about cilia in a minute, but they're these hair-like structures on the surface that allow for sweeping. But these cilia are being pictured in these 3 different micrographs. One is coming from one of the 3, they're each coming from one of the 3 microscopes that we've just discussed. So if you want to play along, pause the video for a second and think about what it is, what each one is. And let's go over the answers. This first one, picture A, we can see inside of the cilia. You can see these linear structures. Those are microtubule structures, which we'll also get to. We see linear structures inside of the cilia. So we're looking inside. We can rule out scanning. So I'm just going to write a note here. We can see inside. This second one, B, we can see the surface with high detail. And then comparing A to C, we can see the cilia here. It's kind of fuzzy. So it's low detail, and we can see nuclei. That means we can see inside of the cells. So this is inside, but low resolution, low detail. So that means A is coming from a transmission electron microscope. B is coming from a scanning. And C is coming from a compound light microscope. So once again, throughout this course, we'll be looking at different pictures, and I'll quickly quiz you about what kind of picture you're looking at. And of course, this will show up on one of the practicals. It'll be important to know what kind of microscope you're looking through so you have an idea of size and detail. And sometimes things look a little bit different using different microscopes. All right, so now we know that cells, we need microscopes, of course, to see them. The cells that we've been looking at, sometimes we see little blobs that look like this. Sometimes they're rectangular with neurons that are really branched. Cells come in a huge variety of shapes and sizes, even in our own bodies. Despite that, despite coming in many different shapes, leading to many different functions, and we'll go over, that's what a lot of this course is, looking at different cells and what their functions are. Despite having so many different shapes and so many different functions of cells, most human cells have the same basic structures. Most human cells have the same basic organelles. When you look in a textbook, you'll usually see this blob of a cell. It's a generic looking cell. Not every cell looks like this. We just saw in the previous slide, some cells are really long, some are really branched. But regardless of the shape or size of the cell, most human cells have the same things. So we're going to go over almost all of these, not all of them, but almost all these different structures here and see what these things do. What are the basic components that every cell needs to exist and be alive and function normally? So one of the most basic components of a cell is the fluid inside of a cell. If there needs to be something to hold it up, to provide a medium for transporting stuff in and out. It's not air, it's a fluid, and it's mostly water and nutrients and wastes that you also want to get rid of. The term for this is cytosol. Cyto means cell. Sol is referring to the solution. Solution, not like an answer, solution like a liquid mixture. That's the cytosol. We introduced the term intracellular fluid before. That also applies here. Intracellular fluid can also mean all of the fluid, all of the cytosol of all the cells in your body, but that can also be synonymous here. Another word you may see synonymous, like what's shown here, cytoplasm. Cytoplasm and cytosol are slightly different technically. Cytosol plus organelles besides the nucleus equals the cytoplasm, but we're going to be loose with that definition. We can also use cytosol and cytoplasm interchangeably, but in case you want to know the nitty-gritty definition, that's what it is. So shown here, this is cells taken from the inside of someone's cheek. You can just scrape the inside of your mouth and create this. I have a video of that. You can see individual cells here. So I'm drawing one cell. Here's one cell. Everything that's not the nucleus, this dark thing that I just colored in is the nucleus. Everything that's not the nucleus inside of the cell, that's cytoplasm. The fluid is cytosol. What's surrounding, let me back up actually, what's surrounding the cell, what's creating a border, that's the plasma membrane. The plasma membrane regulates what goes in and out of the cell. If you've got a little city and you've got a wall around your city, this is like your security gate. This is your city wall. If you have a factory, this is the gate to your factory. You want to regulate what goes in and out. You want employees to come in. You want materials to come in. You want to ship things out wherever you're making. You don't want people who are unauthorized to come into your factory to come in. You want to keep those people out. With a cell, you want to take in certain nutrients. You want to take in certain amounts of water. You want to take in, you want to get rid of waste, but you don't want to let in certain things, like you don't want to let in viruses. You don't want to let in toxins. This plasma membrane helps us do that. We've seen the word membrane before. It's going to come up a lot. Membrane just means a flimsy wall of a thing. Plasma, we've also seen before, but this is a completely different context. Plasma, just the straight up word plasma, is referring to the liquid of the blood. Plasma membrane is referring to the outer covering of the cell. What's really important, well, there's lots of really important things about a plasma membrane, but what's really important about it is that it's semi-permeable. To be permeable means you can let things in and out. Semi-permeable means you can allow only some things in and out. We won't get into too much about the chemistry of this, but this molecule that you see down here, this phospholipid, the plasma membrane is primarily made up of these molecules called phospholipids. As the name suggests, it's a lipid. Fats, for example, are a type of lipid. These lipids form a double layer. They form a double layer. It's called a bilayer. And because it forms this bilayer, some things can go through, but then some things can't. Some things like certain other lipids can pass through. Things like charged molecules or things that are too large will just bounce right off and will not go through the membrane. So it is semi-permeable. You can regulate what goes in and out. Like I said, the major component of plasma membranes are phospholipids. They form the barrier and they selectively transport what can go in and what can go out. Some things chemically are allowed to pass through, some things chemically are not. Other things you can find is another lipid, cholesterol. Cholesterol is a lipid. Cholesterol gets a bad rap. It is true, if you do have too much cholesterol in your diet, that can lead to cardiovascular problems. But cholesterol is a really important molecule that incorporates itself into the plasma membrane. The plasma membrane without the cholesterol is really, really fluid, really wavy. That's why the word plasma is in there. Think about like the surface of the ocean. If the surface of the ocean is too wavy, the surface will break. That's fine with water, but if the surface of our plasma membrane breaks, then everything inside is going to spill out and our cell's going to die. We don't want the plasma membrane to lose integrity. Cholesterol kind of holds things together. It provides some rigidity to the membrane. It maintains the fluidity, prevents it from being too fluid. So that's the goal of cholesterol. That's why it's so important. And then what's pictured here in blue, and there's lots of these things here all over the place, these are proteins. Protein is another class of molecule. Lipids are one type of biological molecule, and they have lots of different functions. And then there's another group of molecules called proteins. And these, the ones that you find in plasma membranes, they can have many, many, many different functions. We're going to go over two right now. What can proteins do for plasma membranes? One thing they can do is act as receptors. A membrane receptor receives signal molecules. Maybe you've heard of hormones or neurotransmitters. The way these molecules act is that they travel through some area, like in the blood or from one neuron to another or something like that. which we'll go over the specifics of that later. But you get a release of this signal molecule, a hormone or a neurotransmitter or some other kind of signaling molecule, and they attach to a receptor. So in this picture over here, this is the receptor, the green thing, and then this blue thing is a signal molecule. When the signal molecule binds to the receptor, it's kind of like a lock and key, a little more nuanced than that, but like how a key fits into a lock. The key fits into the lock and then you can open the door. Something happens when the two meet. If this signal molecule binds to the receptor, kind of like a key into a lock, then you cause some kind of change in the cell. What kind of changes does that cause? It can cause a lot of different things. That's something we've discussed in physiology. It could change enzyme activity, so chemical reactions. It could change transporting, which I'll show you here in a second. It could change, what am I missing? Transporting, chemical activity. And there's probably one more that I'm forgetting, but those are some major things it could affect. In this picture, this animation over here, what we have is insulin. Maybe you've heard of insulin. Insulin is a hormone that helps regulate your blood sugar. Glucose, sugar, it's a type of sugar. Glucose is really important for cells, but it can't get through the membrane. Glucose on its own cannot get through the plasma membrane. What it can do is that if you have insulin bind to a receptor, so this is a receptor right here, the activated receptor causes changes, and then we add channels, these special membrane proteins that allow glucose to come in. That's one thing a receptor can do. We can tell the cell to add these special channels to our membrane. That actually brings us to our second important protein. Receptors are one protein that membranes have. Another are transport proteins. Transport proteins are proteins in cells that allow something to pass from one side of the cell to the other. Could be into the cell, could be out of the cell. I just said that some molecules, like glucose, cannot pass into or out of the cell, at least not through the phospholipid bilayer. These types of things are called non-penetrating solutes. Glucose, things like calcium, sodium ion, potassium ion. These things cannot go through the phospholipid bilayer, but we need them to pass through. To allow them to pass through, we need these transport proteins. So those are two things that membrane proteins can do. They could act as receptors, they could act as transport proteins. There are more things, and we'll get to that when we get to that, but those are two important ones to know for now. So in the next video, where we just discussed cytosol and plasma membrane, in the next video, we'll go over the internal organelles. So let me know if you have more questions. Look at the discussion, rewatch this if you need to. Remember, you can slow this down, turn on the subtitles, let me know if there's any issues with those. Yeah, so I'll see you in the next video. Take care.